In our survey of life, we start at the very beginning, the beginning of life, that is. And so the first cells arose a long, long time ago, 3.5 billion years ago. And there's a lot of debate about how that happened. And so these are all just theories because obviously we can't know exactly what happened, but this is one possible explanation. So scientists think that there was the early environment of Earth had liquid, the atmosphere was low in oxygen, but there was a lot of water vapor. There was a lot of nitrogen containing compounds that had been emitted by volcanoes. There was a lot of CO2. There was methane, which is CH4, so carbon as well, ammonia, nitrogen containing, and hydrogen gas. So these compounds are important because they contain carbon, oxygen, hydrogen, and nitrogen, which you may remember as the 4 elements that are the largest components of living things. And so you had this sort of stew that contained the compounds of life. You also had energy coming in from like lightning strikes and things like that in this really sort of volatile atmosphere of early Earth. And so they think that may have initiated the combining of these small molecules, again, the carbon dioxide, the water vapor, etc., starting to join into larger molecules that we call macromolecules. And then the idea that the next step was that these large molecules started to get wrapped in other large molecules, and specifically ones that were amphipathic, that is, containing both polar and nonpolar regions. These would have been protomembranes. And so once you had this surrounding, that created the opportunity for there to be a difference between the inside and the outside. And once you have those differences, then evolution can really take off from that point. And then kind of an extra piece onto that was that these self-replicating molecules formed. So self-replicating molecules are things like DNA and RNA, and a lot of people think that RNA was probably around first. And remember, we consider it self-replicating because it's got those base pairs that when you make those base pairs, you can make a match to an existing molecule. And so again, this is like a possible theory and sequence for how we would have gotten these very earliest cells. So in the fossil record, we first see evidence of these early simple cells about 3.5 billion years ago. So remember, Earth itself is 4.5 billion years old. So 1 billion years after Earth forms, we start to see these early cells. So this is the evidence for this is a lot of times in just the evidence of the activity of these cells. So this is a picture of stromatolites. These are modern day ones. They're mineralized structures that form from the metabolic activity of certain types of bacteria and archaea. And so we see fossil evidence of these stromatolites in the fossil record. And so we know there must have been prokaryotes around making them. We can also see imprints in sediment of bacterial cells, again, at the microscopic level. So if you look at these fossilized sediments with a microscope, you can see the imprints of some of these cells. So that's where we get this 3.5 billion years ago number. So the modern descendants of the earliest cells are the bacteria and archaea, are their most close relatives. And these prokaryotes are everywhere. They're mostly microscopic, so we don't see them, but they're present in very large quantities. And so by weight, they're the most present organism. If you looked at, for example, a handful of soil, there's more prokaryotes in that soil than the number of humans that have ever lived in the history of Earth. One of the reasons why they're so abundant and prevalent is that they're very, very adaptable. And so in environments where you can't find animals, for example, environments that are too acidic, too salty, very, very cold, or very, very, very hot, these kinds of environments tend to limit most other organisms, but prokaryotes can still thrive there. So how many types of prokaryotes are out there is a really hard number to know, because for the most part, we have not seen any of these prokaryotes. We haven't studied them. And that's because we're limited by techniques. So when we go to grow bacteria in the laboratory, we have certain conditions that we can easily replicate for them. And so that limits which species can grow. So we estimate that probably only 1% of species are easily cultured in the laboratory. And so again, that makes it really difficult to study them because you just can't get the quantities that you need in order to really look at them or grow them or study their needs. And so in many cases, we might only know a prokaryote by its DNA sequence. So there are scientists who actually go into an environment like the ocean and take out samples and look for unique DNA sequences in that sample. So they're not seeing cells, they're not seeing anything about this organism. All they are seeing is its DNA sequence and being able to say, yes, this is a distinct DNA sequence. No other organism has it, so this must be a different type of bacteria or archaea. So bacteria and archaea are our prokaryotes. So remember, we use the term prokaryote in comparison to eukaryotes. Eukaryotes contain or have subcellular compartmentalization in the form of organelles, membrane enclosing structures that separate functions of separate the functions within a cell, whereas prokaryotes do not have organelles to separate the functions of the cell. They tend to be smaller, eukaryotes tend to be larger. The most iconic organelle is the nucleus, so sometimes people narrow this down to say, okay, eukaryotes have a nucleus and prokaryotes don't, but remember that that's a simplification. Eukaryotes have organelles, including the nucleus. Prokaryotes do not have organelles, including not having a nucleus. And so when we look at the history of these organisms, so let me go over here a little bit. We have ancestral cells. And so way back in time, we get a divergence of those ancestral cells into modern day bacteria, or that would give rise to modern day bacteria. And then we have a separate lineage that comes from that very early split. And that separate lineage will give rise to archaea and eukarya. So remember that the top of my tree is my modern day organisms. And again, this is way back in time. So we have two splits that give rise to our three major lineages of organisms. And so one of the things I want to make sure to point out about this lineage is what this suggests is a more recent common ancestor for archaea and eukarya. So archaea and eukarya are more closely related to each other than either are to bacteria. And this surprised scientists and of course most people as well, because when you look at bacteria and archaea, they're both prokaryotic. So people assumed that being prokaryotic meant they were close relatives. Okay, so that's not true. Even though we consider bacteria and archaea to both be prokaryotes, again, I want to reiterate that archaea and eukarya actually share a more recent common ancestor and are therefore more closely related than either are to bacteria. So the further implications of this cladogram is that the eukaryotic nucleus and cytoplasm arise in this shared in a shared ancestor or an ancient archaeal organism. And so, let me say that again. It does not arise in this shared ancestor. It arises in the lineage that branches from that shared ancestor, just to be really clear. So if I was to draw this into a cladogram, I would have nucleus and organelle here. So this is a derived characteristic, to use our language from the previous chapter, that arises in eukarya. But again, this is a close relationship between archaea and eukarya. So an archaeal organism gave rise to eukarya. So another sort of interesting thing about the eukaryotic lineage and how it relates to prokaryotes. So even though eukaryotes are deriving from an archaeal ancestor, there's actually a movement or an evolutionary relationship of bacteria to eukarya, and this has to do with the semi-autonomous organelles, the mitochondria, and the plastids, and we could say more specifically chloroplasts, so it applies to some of the other plastids as well. And that is two types of bacteria, proteobacteria and cyanobacteria, colonized the inside of the primitive eukaryotic cell. So at some point after this split, there was bacteria who started living inside of early eukaryotic cells. This is called endosymbiosis. And so these bacteria would live independently inside of this early eukaryotic cell for thousands and thousands of years, presumably. But of course, today, that's not true. They're not actual bacteria inside of it. They're the mitochondria and the chloroplast. And so you learned about this in a previous class, but just to remind you, mitochondria and chloroplasts are derived from this ancient symbiotic relationship. So again, it's called endosymbiosis. It's when a smaller species lives inside of a larger species. And this is actually inside of the cell. So what we think happened is that we had a proteobacteria that moved into the inside of this early eukaryotic cell. And then over time, this independent bacterial cell would stop being independent. So it would lose a lot of its genes to the nucleus and no longer be its own cell, but it retains some traits that show us that it was its own cell at one point. So for example, it has its own DNA. So this applies to the semi-autonomous organelles, to use the term that you learned in the previous class. They have their own DNA, and they are self-replicating. Oops. Get a little neater here, self-replicating. And so that's what makes them semi-autonomous. And again, they have their own DNA and they're self-replicating because they derive from this bacteria that went inside of them. And the same thing happened with the chloroplast. So in plants, you had both a proteobacteria and a cyanobacteria that colonized the inside of the eukaryotic cell. And this would eventually give rise to plant cells that contain both types of chloroplasts over long periods of time. So again, that's explaining this line across here. So again, eukarya derived from an archaeal ancestor, but we have transfer of proteobacteria and cyanobacteria to the eukaryotic cell through endosymbiosis.